is 15 nanometer by 20 nanometer. This is the dimension. The prokaryotic ribosomes, they are associated with the plasma membrane. And mostly what happens in the case of the prokaryotes, since they do not have a nuclear membrane, so all of their structures are just lying in the cytoplasm only. So ribosomes also lie in the cytoplasm. Now the type of ribosome present in the prokaryotes, it is called as 70S kind. In us, we have 80S type of ribosome, and this S here is the sedimentation coefficient. Okay, it is the degree by which the ribosomes were studied at that point of time. So they are of 70S type. And this 70S consists of two subunits. One is the larger subunit and the other is the smaller subunit. The larger subunit is 50S and the smaller one is 30S, and altogether they are 70S. Don't count. Okay. When you count it, it does not come out to be 70S. It's just that their weight is 50 and 30S, meaning how it sediments, how it settled down at the bottom. Now, in the case of prokaryotes,
Ma'am,
what happens? Yes?
ma'am, you told that they won't have ribosomes, right?
No, they will. This is the only cell organelle that they do have.
Oh, okay. Okay.
The reason being, this cell organelle does not have a membrane bound structure. Like it does not have a membranous structure. That is why. So since it
Okay.
doesn't have a membrane, so they'll have it. If
Okay.
ribosomes would have had a membrane present, then the prokaryotes couldn't
Then,
have had ribosomes. Now, when many ribosomes attach themselves on mRNA, okay, mRNA is a type of RNA. M stands for messenger. So mRNA is messenger RNA. So there is a structure that these ribosomes form, is that like mRNA is like this. Okay, it is a strand of RNA and many ribosome come and attach onto it. Bigger subunit, smaller subunit. Bigger subunit, smaller subunit. So many ribosomes attach themselves on the mRNA, and this structure is called as polyribosome. Poly means many, and they are forming sort of like an aggregation. So we call it as the polyribosome or polysome. Clear? Now talking about the function. So the function of this polysome or the ribosomes that are present as a polysome is to translate mRNA into proteins. See, the protein formation is known as translation. Okay. In our like biological system, the formation of protein in terms of molecular biology, it is called as translation. So from mRNA, the proteins are made. I'll tell you one thing in advance, like this is not to be known by now, but see, DNA, like from the DNA, mRNA is formed by the process called as transcription. Transcription. And then from the mRNA, the proteins are formed from the process called as translation. Okay. So that ribosome or that polyribosome, they perform here.
so, ma'am,
Why? Hmm?
ribosomes, I mean, like prokaryotics, they have DNA present in them.
Hmm. They do.
Okay. So, they have like DNA present in them because they are coming from mRNA.
Yeah, they do have a DNA. They have mRNA also. They have proteins also. But they do not have membrane bound nucleus, meaning their DNA is scattered in the cell. It's not bounded by a membrane. Clear? The whole entire
Yeah.
ribosome is clear.
Yes, ma'am.
Then comes inclusion bodies. Now see, inclusion bodies, they are again non-membrane bound structures. So they can be easily found in prokaryotes. And these non-membrane bound structures, they are present in the cytoplasm. Now what happens? Inclusion bodies, imagine them being like a storeroom for the bacteria. It's a storeroom. What does it do? It's like it stores the reserve food material. Okay, all of the reserve food material in the case of bacteria, it is stored in the form of inclusion bodies. Now talking about the inclusion bodies, so these are the examples, like they have phosphate granules, They have synovial granules, glycogen granules, meaning granules means particles like these. And inside of them, certain stuff is stored, like phosphate is stored, synovial granules stored, synovial stuff. Then glycogen is also stored in them. So these are vesicle-like structures which store these stuff. Yeah. Then comes, like in the case of blue-green algae or cyanobacteria, like you have studied, in them, and in the case of purple and green sulfur bacteria, they have gas vacuoles, because for buoyancy, they require something to lift them up because they are mostly in the aquatic environment. So they need gas vacuoles. So remember, vacuoles and inclusion bodies, they have a very similar function, that they are responsible for storage properties.
Gas vacuums in the sense they store.
See, gas vacuoles, they store air,
Yeah.
right? So they help in buoyancy.
Okay.
Whereas the rest of the vacuoles, they store certain chemicals, which is sort of like a reserve food material storage in the case of bacteria. Got it?
Yeah.
Then comes the eukaryotic cells. Now, eukaryotic cells, we know that they will have a proper membrane bound nucleus. All of the membrane bound organelles will be present in them. The protists that you have studied, the fungi, the plants, the animals, all of them are eukaryotic organisms. Only bacteria are prokaryotic. Like all the monerans are prokaryotic, but rest of them are eukaryotic. What kind of members protists are?
Like,
Like, if I say to tell me about protists in 2 words, what would you say?
they're like
They are
multicellular,
unicellular eukaryotes.
multi-organisms. Sorry,
They are unicellular eukaryotes.
your mic,
Am I
ma'am, is
audible?
it? Yeah, ma'am, is it like yeast, right? But in
Yeah.
protists form.
Yeah, they, like all of the members of protists, they are unicellular eukaryotes, but only yeast is the only member of fungi that is a unicellular eukaryote. Now, the cells which possess a membrane bound nucleus along with membrane bound cell organelles are called as eukaryotic cells. A proper plasma membrane is present. Cell wall is also present, but we can find them only in the case of plants, fungi, and some protists. Cell wall is completely absent in the case of animals. What is the plant cell wall made up of?
Yeah, cell wall, cellulose,
Fungal cell wall.
chitin,
Okay. And bacterial cell wall?
Indian cell,
Starts with a P.
polysaccharides,
Peptidoglycan.
peptide,
Peptidoglycan.
peptidoglycan. Okay.
Now, coming on to the differences between the plant cell and the animal cell. Right now, we know that the plant cell, in terms of size, the plant cells are larger than animal cell. Okay, and the animal cells, they are generally smaller in size. Plasma membrane has another layer, that is cell wall, which is made up of cellulose. But in the case of animals, plasma membrane is the outer layer and that's it. No cell wall, no additional layers are present. In the case of plant cells, plastids are present. Plastids, I'll tell you later on. Like there are 3 kinds of plastid: chromoplast, chloroplast, and leucoplast. So those are present, and those plastids are absent, obviously, in the case of animals.
But
Now,
ma'am, we
hmm,
also have like we also have plastids, right? Because do we like
See, I'll tell you, plastids, like one of the plastids are responsible for having chlorophyll in them.
yeah, no, but like we can have the other two, right? Lycoplast and if no,
How? Why? We don't need it.
okay.
Then most mature plant cells have a permanent vacuole and a large central sap vacuole. So, like in the case of plants, the vacuoles are very big in size, okay, and they are prominent and permanent, meaning they'll stay there in the case of plants. But in the case of animals, the vacuoles can be present, and whenever they are present, they are temporary vacuoles. No permanent vacuoles are found in animal cells because animal cells are always dividing. And apart from that, most of the time, the vacuoles, they are not present at all. It's absent in the case of animal cells. Now, Golgi apparatus of the plants, they are simple and we call them as dictyosomes, whereas the Golgi apparatus of the animals, they are highly complex. So, Golgi apparatus is called as Golgi apparatus in animals, but we call it as dictyosomes in the case of plants. All of these things you will study later on. Now, plant cells do not have the centrosome and the centrioles. They lack the centrosome and centrioles. These two structures, they are like important for cell division. So
Okay.
plants don't have it and animals do have it. Clear? Now
I have a
starting,
doubt. So plants don't do cell division?
they do, but they have different mechanisms.
Okay.
They do all the kinds of cell division, but they have a very different mechanism from us.
So they won't have dead cells like no, they have dead cells, but then they don't do that cell
They
division.
have different mechanism. I'll teach you that in the next chapter, how the animal cells and all division, how it takes place, because it is not very simple. It's like a very elaborate process. To explain you one thing, I'd have to explain you the whole entire thing. Then only you will understand how the division in plants take place. Okay. Now, talking about the plasma membrane, or you can also call it as the cell membrane. Now, it is the limiting membrane of the cell. Okay, limiting membrane meaning the outermost layer of the cell, and the invention of microscope in the year 1950s, it enabled us to understand the structure of plasma membrane better. Initially, people gave theories about how it would be like, but when the microscope got invented, then it was understood in a very clear picture way. Now, the structure of plasma membrane, it was described by Singer and Nicholson, very famous. Like they are famous for giving the fluid mosaic model. Okay, Singer and Nicholson, they gave a model called as the fluid mosaic model, which explained the structure of plasma. And they did it in the year 1972. Now, see what happens is that plasma membrane, it first of all you understand the two things, hydrophobic and hydrophilic molecules. Now hydrophobic molecules, they are water hating
They
molecules.
don't like water. They're
Yeah,
phobic, they hate water.
yeah, they hate water.
Metrophilic are the ones which like water.
Yeah, they are water loving and these are the water hating. So the molecules that are water hating, they'll try their best to stay away from the water.
Okay.
So as a protective mechanism, they'll like the, for example, if this is the hydrophobic head, hydrophilic tail.
The inside will
So
be
yeah,
hydrophilic.
so it will be present towards the inner side. Got it?
Yeah.
Hmm. Apart from that, remember that the structure of the plasma membrane is a lipid bilayer, meaning it is made up of two layers of lipid. Lipid means fats. So the plasma membrane is a fatty layer which has two fatty layers. Now, in the lipid bilayer, there is the presence of proteins. Okay, proteins are embedded in the lipid bilayer, and these proteins are of two types depending upon the ease of extraction, meaning how easily we can take out the protein. So if we can take it out very easily, so that is peripheral protein, meaning present on the periphery, lying on the surface, so you can easily extract it. And the integral protein, they are situated deeper inside, so we can't like extract it very easily. So they are embedded but partially embedded. So these are the two kinds of proteins: integral proteins and peripheral proteins.
Ma'am, I have a doubt. So
Hmm.
when the water goes, it can't enter, right? Because there are hydrophobic um,
No,
um,
see,
things.
water
So
does not necessarily go
on
inside
the
the
mic,
plasma
mic, the
membrane.
mic,
Huh?
the mic.
Oh, I'm saying the water does not necessarily go inside the plasma membrane as such, but these hydrophobic molecules, like, they are present inside like this, and they are protected by the hydrophilic molecules. So nothing happens to them. They never come in contact with the water. The part that will be coming in contact will always be the hydrophilic moiety.
Um,
Okay.
so, mm, like the
Now
lower
talk.
part
Yeah.
also gets the water.
Hmm.
The lower part, the lower hydrophilic also gets the water.
Yeah.
How is the water transpiration going?
See, see, in the case of the plasma membrane, there are certain channels, ion channels, because of which the substances can move through, like, to and fro. Okay, it doesn't necessarily,
Like,
don't imagine them as having like a, wave of water coming and going. That is not going to happen. It's a very regulated manner structure that happens in the plasma membrane. Okay.
ma'am, is it
Hmm.
because like there are like proteins and everything over there, so those things will take in water and maybe that's
See,
why?
there's no taking in of water or giving out of water. It's just that their nature is hydrophobic. The nature.
Yeah.
It
Yeah.
doesn't have anything to do with the water. Like, it doesn't mingle around with water. It's just the nature that they have.
Mm, okay.
Okay. Now talking about the fluid mosaic model. So this was given by Singer and Nicholson. And according to this model, the plasma membrane have a quasi-fluid nature or a semi-fluid nature. Okay, it's like always moving, and uh, the like since the plasma membrane has a semi-fluid nature or the quasi-fluid nature, because of this only the protein that I told you about, the integral protein, the peripheral protein, they can move to and fro in the plasma membrane. Why? Because the lipid bilayer has a semi-fluid structure, and the protein that are embedded in them, they can move because it is fluid. Talking about the functions of the plasma membrane. So the fluid nature of the plasma membrane is important from the point of view of functions like cell growth, secretions, endocytosis, cell division, etc. Meaning the fluid nature of the membrane allows it to perform all of these functions. Cell growth, secretion, endocytosis means it is the intake of certain materials inside the cell membrane. It involves the intake of materials in the form of carrier vesicles formed by invagination of plasma membrane, meaning the plasma membrane again deepens like that and forms a vesicle inside. So that is endocytosis. Okay. So, like, while we are discussing, we should also discuss about exocytosis. So exocytosis is just the opposite of endocytosis. Here, the vacuole or the structure that is formed, it is excreted out of the cell through that plasma membrane.
I didn't understand.
I'll tell you.
Endocytosis is like what is
For
endocytosis?
example, this is a plasma membrane, okay, surrounding the cell. Now, whenever there is the need of any kind of molecule to come inside the cell, meaning the cell wants to have it. So the plasma membrane will start deepening like this, will start deepening from both the sides like this. And then it will pinch off and found like form a vacuole or a cell. So now the stuff, whatever was supposed to come inside, now is present inside this.
In
This
the
is
form of
endocyte. Yeah, in the form of certain vacuoles or vesicle-like structures. And
Okay.
when the cell needs to excrete it out, so this one will start coming here again, forming, sort of like fusing with the plasma membrane, and this thing will be excreted out.
Okay.
Got it? So this is exocytosis. You can also call it as cell vomiting or effage. Apart from that, the plasma membrane allows the transport of materials across it, because the nature of the plasma membrane is selectively permeable. It is permeable, but only to selective molecules, not bigger molecules, not charged molecules, some smaller molecules. Now, the cellular transportation, like inside the cell, there are various kinds of transportation that take place. The cellular transportation, mostly it is of two types: passive and active transport. Now, passive is the one which does not require any energy, meaning here the movement of substances take place from a region of its higher concentration to the region of its lower concentration. That is along the concentration gradient, high to low, isn't it? For example,
Yeah.
if you're standing at the top of the hill and now you want to push a big rolling stone, it will be much easier for you,
Yeah.
isn't it? Coming from a high to low area. But now pushing it from the low area to the high area, it would be so difficult, isn't it? It would require energy. So passive transport, remember, moving from high to low, this is called as along the concentration gradient. Okay, along it. See, I have drawn some. I think these kind of figures are there in physics, isn't it?
Yeah.
Hmm. So passive transport is the movement of molecules without any energy requirement. Because it is happening along the concentration gradient, it does not require any energy,
Mm.
any
Oh, yeah,
external
because
energy.
it's anyways going from high to low, so it doesn't
Yeah.
require.
Why would it need? Like if the rolling stone is coming down a hill, would you go and push it? That go faster.
Mm.
It'll already go faster. Even you
Yeah.
will be running faster, isn't it? So that's the case. Now talking about diffusion. So diffusion is mostly the movement of gases along the concentration gradient through a semi-permeable membrane. That is diffusion for us. And the same thing, meaning moving from high concentration to low concentration, when it is performed by water, meaning a liquid. So we call it as osmosis. Water also follows the law of diffusion, but the diffusion of water is called as osmosis. Got it? Now, neutral solutes can do passive transport. Neutral solute means the ones that do not have any charge. They are neither positive nor negatively charged. So they are neutral. Neutral means no charge. So all
Also.
those molecules that have no charge on them, they can easily pass through the passive transportation without the use of energy. But
Ma'am,
when
what
we,
charges? Like,
huh?
what kind of neutral charges?
Charges, like for example, if I say sodium, sodium has positive charge.
Yeah.
If I say chlorine, chlorine has negative charge. So these are
Okay, like that.
ions.
Yeah.
Okay, ions can't. Charge doesn't mean the phone charge. Got it? Here, charge refers to the positive charge or negative charge. Got it?
Yeah.
Then comes the next type, which is active transportation. It is also known as uphill transportation. Again, now in this case, the movement will happen against the concentration gradient, meaning from low to high. Now you would require energy, and that energy comes from ATP, which is the adenosine triphosphate. Okay.
Oh, we're coming here to mitochondria. So
Hmm,
what?
no, not yet.
Never
But
mind.
yeah, we are close. We are close. And this is an energy dependent process. Why you have an affinity for mitochondria? Mitochondria
What?
fascinates you.
Yeah, in some way, I don't know. Because it's like the powerhouse of the cell, right? It gives out the ATP. That's
Okay. Now the active transportation, it is an energy dependent process. dependent. It is an energy dependent process. And the best example is sodium potassium pump in the case of animal cells. Meaning now you can see how charged species are here, isn't it? They have positive charge. There are certain that have negative charge. So for their transportation, active transportation is required. Clear?
Mm.
Any doubts in plasma membrane?
No.
Anything that troubled you?
Mm. Ma'am, the hydrophilic and the yeah, that part. I have another question. Just the last one. So
Mhm.
in that, so like in between them, they will have other things also, right? So it won't be completely hydrophilic and phobic over there, correct?
It's a combination of both. Like they are present as a unit.
Yeah, in between those, there will be other molecules also, right? So that's how they can perform osmosis also, correct?
Yeah.
And even diffusion can take place like that. But
Yes.
I have a question again in that. So in diffusion part, they don't like water and they like water. So air can be passed through easily, right?
Air.
Yeah.
Air is gas only, right? So
I mean, the
diffusion.
gas only it can go, right?
Diffusion.
Yeah, diffusion can happen easily, right? Okay, that's all. Yeah.
Oh. Now talking about the cell wall, we know that the cell wall is present in plants, bacteria, fungi. Animals do not have it. Now the plant cell wall made up of cellulose, which is a polysaccharide. Poly means many, saccharide means sugar. So polysaccharide is a complex sugar. Have you studied biomolecules yet?
All
Now
I've
the
heard is
plant,
the chapter, that's all.
huh?
All I've heard is that it's that a tough chapter, that's it.
It's not. We'll not make that mindset beforehand.
Mhm.
Okay. Even I did not used to like biomolecules while I was studying it. But once I've started to teach the chapter, it is easy. It's nice. Not easy. Obviously, it will be easy. I'm past that class, but it's interesting now. So it's not boring and it's not difficult. So don't worry. And I'm here. So don't worry anyway. Even if it is difficult, we'll do it together. All right. Now the plant cell wall, it is cellulosic. And apart from cellulose, the plant cell wall may also contain all of these substances. There is hemicellulose, pectin, proteins for extra protection. Okay. Then comes fungal cell wall. We know that it is made up of chitin. So we'll call it as chitinous cell wall. Algal cell wall, which is in the case of algae, they are composed of cellulose, galactins, and mannans. Their cell wall is also cellulosic. But apart from that, they have extra stuff like galactins and mannans. But the plants, they have hemicellulose, pectin, proteins, and all extra structures.
Ma'am, what is hemicellulose?
Hemicellulose is a form of a cellulose only with certain structural differences.
Oh, okay. So they compose of cellulose and other things also. Okay.
Like the chemical composition is different. Now, the plant cell wall, it has two regions, primary cell wall and secondary cell wall. See, primary cell wall is the cell wall that is formed first, meaning like while the cell wall development is taking place, the cell wall that is formed first will be the primary cell wall. And then after that, there will be the secondary cell wall laid. Okay. Talking about the primary cell wall, it is the first formed cell wall of plants. It is thin and expandable.
Mm.
Most of the young plants, they possess a proper primary cell wall. This will be soft also. Okay, it will be soft and it will be thin. Now, in the case of mature cells, they will have one more layer of wall, that is secondary wall, and it is thicker than the primary wall. Now, in between the primary and the secondary wall, there will be a cementing material, which will be the middle lamella to
Ma'am,
join them
how are
together.
the ma'am, how are the first and second? um like attached?
Middle lamella.
Oh, okay, okay. Because of that. No, no, like how are they like arranged?
See, for example, if the cell is forming, first the primary cell wall is made.
Yeah.
During this time, the cell is young. But now when the cell matures, then another cell wall will be arranged.
Oh,
That
on
will
top
be the
of the
second.
first one.
Yeah, and in between them, the middle lamella will be present.
Mm.
So middle lamella, it is the cementing material or sort of like a gluing material to keep the wall layers together. It is very important that it is made up of magnesium and calcium pectate. Clear?
Mm. Yeah.
After that comes plasmodesmata. Now see, plasmodesmata, they are sort of like a bridge. For example, this is a cell wall, this is a cell, and this is another cell. So in between two adjacent cells, there will be a bridge-like structure, which is the plasmodesmata. What is its function? It connects the two cells together. Sort of for cell-to-cell communication, plasmodesmata is there. Okay, all right. For cell-to-cell communication.
Ma'am, so there will be a gap between the cells.
Yeah, it will not be this prominent, but there will. Like this
Sort of
is
like
just a pictorial
synapse.
representation. Like for
Mhm.
example, maybe the cells are situated like this, but maybe there is this connection in between them.
Okay.
It depends from cell to cell. Okay.
Mm.
Then comes the functions of cell wall. So it maintains the shape of the cell. It protects the cell from mechanical injury. It like again, it keeps the cell free from bacteria, viruses, meaning any pathogen. And it allows the material to pass in and out of the cell. It helps in cell to cell interaction. Okay. Apart from that, it performs one more thing. It does not let the cell burst or shrink. Clear?
Yeah.
Any doubts up until now?
Mm.
Okay, so you note down till here. I'll give my throat a rest in the meantime. Um, you have to note down from prokaryotic ribosome, right? Okay, you can note it down. You can switch off your video. Lie down comfortably.
Can you scroll?
Yes.
Ma'am, could you show that part where you told DNA becomes into mRNA? Yeah, thank you. Yes, ma'am. Thank you, ma'am. Ma'am, can you show from inclusion bodies? Okay. Yeah. Mhm. Mhm. Mm. You can show the protein part depending upon the ease of extraction, right? Yes, ma'am. Yes, ma'am. Mm.
Done till here? Yes, Rida, done till here?
Ma'am, I can't hear you.
Are you done till here?
Yes, ma'am.
Yes, okay. So we'll keep it till here only for today's class, okay? And we'll continue the rest in the next.
Yes, ma'am.